Biology, Chapter 19, Theory Video 6. We are going to discuss about promoters. So now, just like the humans or all the other organisms, bacteria also contain many different genes, and they produce many different proteins. But not all of these genes are switched on at once. They are going to produce proteins that are required in the conditions in which they are growing. So whatever is suitable for their surrounding and whatever is suitable for their existence, they produce. Now, we have seen earlier that E. coli will enzyme enzyme beta galactosidase only when they are growing in a medium containing lactose, and also when there is no glucose available. So both of those conditions need to be there. So the lactose is there and glucose is not available. So when that happens, they produce enzyme beta galactosidase. So this expression, such as in the lactoperon, is controlled by promoter. So that means the expression comes about with the aid of a promoter. So this promoter is defined as a region or a length of DNA that includes the binding site for RNA polymerase, where transcription of a gene or genes begin. In eukaryotes, promoters also have sites for binding of transcription factors, right? So here what happens is DNA, this is going to be the DNA region or the part of the DNA where RNA polymerase will bind when it starts transcription. So if we require the gene to be expressed, we have to, now we know how to add the gene. Now after adding the gene, we have to add an appropriate promoter as well to express this gene. If not, it will not be expressed. So now when the bacteria were transformed to produce insulin, so they had to add it next to the beta galactosidase gene. So they had the same promoter. So the promoter would switch on the insulin gene when it needed to metabolize the lactose. But if it now when the bacteria was growing in a medium of lactose but there was no glucose, they produced both the galactosidase and human insulin to produce whatever they needed, that is the glucose. So the promoter has different functions. It allows the RNA polymerase to bind to the DNA, and also it would make sure, or it would ensure that it is going to recognize the two DNA strands is the template strand. Out of the two DNA strands, what is going to be the template strand is recognized by the promoter. So there is this, in this sequence of nucleotides in the promoter region, you would have the transcription start point. First nucleotide of the gene to be the first nucleotide of the gene is to be transcribed. So that is going to be in the transcription, the transcription start point is going to be identified by the promoter. So in that sense, we can say that promoter controls the expression of the gene, and also it makes sure that a high level of gene expression is established. That is, without the promoter, it would not be, sometimes it might not even be expressed at all, or else it would be expressed but not at a higher standard. Now in eukaryotes, a protein known as transcription factors also would be required to bind the promoter region to RNA. Or to RNA polymerase before transcription can begin. So that means in eukaryotes, there are two requirements. Transcription factors would also be needed to bind to the promoter region or to the RNA polymerase. So I hope you understood. If you have any doubts, please ask me. Write down promoters. There are many genes in bacteria. Not all of them would be expressed. What is expressed are the ones which are required in the conditions in which they grow in. To express a gene, a promoter is required. The promoter is the region of DNA to which RNA polymerase would bind when it begins the transcription. When a gene is added, an appropriate promoter also should be added. Promoter has two functions. Number one, it allows RNA polymerase to bind to DNA. Number two, it ensures that it recognizes which of the two DNA strands is the template strand. I hope you understood. If you have any difficulties, ask.